Alright guys, we're going to go over enzymes today. So what are enzymes? Enzymes are most proteins. They act as catalysts to speed up a reaction, not permanently changed in the process. And then here we have an example of an enzyme. Enzymes are specific for what they will catalyze, which means they will only work on one certain type of reactant. They are reusable, so we can use them over and over again. And they end in ase. So sucrase would break down sucrose, lactase would break down lactose, maltase breaks down maltose. So here we have a picture of, here's our enzyme. An enzyme has an active site which only fits one type of substrate. So how do enzymes work? Enzymes work by weakening bonds, which lowers activation energy. So we don't need as much energy to get the process done. So here's our, without an enzyme, our reaction. Here's our reactants. We need a large amount of energy in order to get to our products. If we add that enzyme, we can do the same reaction, but with fewer energy. So here we have enzymes and reactions. Here's our substrate, hydrogen peroxide. Here's our catalase, our enzyme, which is liver homogenate. Here's the enzyme without the substrate. When we add the hydrogen peroxide, you can see all these bubbles. So the enzyme plus the substrate gives us our water, or H2O plus bubbles. That's our product. So we're breaking down the hydrogen peroxide. The enzyme substrate complex. The substrate, or the substance the reactant and enzyme acts on, is the substrate. So here's our enzyme. Here's our substrate. The enzyme is going to break down the substrate. It's going to join at the active site. So a restricted region of an enzyme molecule which binds to the substrate is called our active site. Induced fit, a change in the shape of the enzyme's active site, induced by the substrate. So we've got our enzyme, and then our reactant would fit right in there. The substrate fits right in there. And then active site is going to wrap gently around it, kind of holding that substrate in, so that way it can break it apart and release the products. A change in the configuration of an enzyme's active site, the hydrogen ions and ionic bonds are involved, induced by the substrate. So induced fit is when we're talking that your active site is going to wrap around that substrate. What affects enzyme activity? 3 factors: environmental conditions, cofactors and coenzymes, and enzyme inhibitors. Environmental conditions. Extreme temperatures are the most dangerous. High temps may denature or unfold the enzyme. So if it gets too hot, those enzymes will not function. The pH. Most enzymes like to be 6 to 8 near neutral. So anything, and not all enzymes are the same. So some in your mouth are going to have a different pH than the ones that are in your stomach. The ionic concentration. So how much salt ions are located in your body also affects how those enzymes function. Cofactors and coenzymes. Inorganic substances such as zinc and iron and your vitamins are sometimes needed for proper enzymatic activity. So if you do not have enough zinc and iron or other vitamins in your body, sometimes your enzymes won't function properly. So an example, iron must be present in the red blood cell. Hemoglobin is used to carry oxygen throughout the body. So if you don't have that hemoglobin, you become anemic. 2 examples of enzymes inhibitors. A competitive inhibitor are chemicals that resemble an enzyme's normal substrate and compete with it for the active site. So we have a competitive inhibitor that's going to try to fit into that active site of that enzyme, but that enzyme really can't break that competitive inhibitor down, so it's slowing that reaction because it wants that substrate. And then non-competitive inhibitors. Inhibitors that do not enter the active site but bind to another part of the enzyme, causing the enzyme to change its shape, which in turn alters the active site. So if we add on that non-competitive inhibitor to our enzyme, we're changing the function of that enzyme, so we're changing that active site. Now our substrate can't fit there because now it can only fit with some other type of substrate. So we've changed that whole function of that enzyme. All right, guys, notes are over.